Okay, so in the interest of time, let's get started. So it's my honor to have a chance to introduce Professor Kang from Howard Hughes Medical Institute at Stanford University. Professor Shen actually did his did his bachelor in Tongji Medical University, and subsequently went to the U.S. and was staying in the U.S. Until now, right? So he did his PhD in Duke, and then subsequently went to UCSF at Corey Bartman's lab for his postdoc. With a few years of postdocs, and started his career at Stanford University. So currently, Professor Chen is the director of the Wuxi Neuroscience Institute. And in recent times, Professor Chen has actually got a number of awards, and I think I wouldn't go into the details, but just to quickly mention, in 2025, just past year, he was elected to both the American Academy of Art and Sciences and the National Academy of Science. So congratulations, Professor Chen. So I think with that, I think I would just let Professor Chen to tell us his research. I apologize, I was running down from another facilities with another visit. So apologize for my kind of not the most smooth introduction, Professor Chen. Yeah. So with that, please welcome Professor Chen to give us a lecture.
Thank you very much, Tom. It's a great pleasure to be here. Actually, I've been here many times, believe it or not. And that's thanks to Nancy's, I don't know if Nancy's here, but she played a big role she's actually instrumental in organizing this GRC, the whole series of Gordon Research Conference in Cellular Molecular Neurobiology here in the Hong Kong University of Science and Technology. And I personally actually organized the 2012 GRC, and lots of vivid memories comes to mind. The fancy seafood lunch at this village, and then jumping into Clearwater Bay, and has never experienced the feeling of jumping into like a bath, a warm bath. Whenever you jump into water, ocean water, you always feel like it's going to hit you with chill, right? But that was really some memory that stuck with me. But of course, I think that GRC has really provided with several generations of neurobiologists, especially from mainland China and from Asia, an opportunity to really meet a lot of speakers. And I thought that was a fantastic activity, and hope that someday we'll bring it back to Hong Kong. But as Tom alluded to, what I want to talk about is neuronal cell biology. And so, of course, we're talking about the neural circuits, and then our brain is a giant neural circuits, right? This neural circuit is assembled through a series of developmental events that starts with cell generation. Mitosis gives rise to neuronal cells. Post-mitotic cells then undergo a series of morphogenesis events, including forming an axon and guiding the axon to the right place, and growing an elaborate dendritic arbor. And as this is happening, the cell becomes a polarized cell, sending different proteins, different organelles to distinct subcellular compartments. And then this is followed by formation of synapse formation, where pre- and postsynaptic neurons touch each other, and then this initial phase leads to the formation of immature synapses, synaptic circuits, and activity-dependent mechanisms then comes in to refine the neural circuits by eliminating and strengthening particular synapses. And you can imagine that this is a very complex process that essentially established this nervous system based on genetic and sometimes activity-dependent mechanisms. And in the past, we have really focused on various questions in this theme. So my lab has been focusing on developmental neurobiology. We started off by trying to understand what are the cues, what are the signals that decide which neurons should form synapses and which should not. And we identified initially, after I went to Stanford, we identified pairs of adhesion molecules that specify, that promote synapse formation. So if those cells have the same pairs, that they will form synapses. But surprisingly, we also found other ones that are inhibitory to synapse formation. There are signals in the environment, including like morphogenetic gradients, such as Wnt, such as natrin. They seem to suppress synapse formation, and then that also contribute to carving out the right connections. And later on, we studied basically the protein aggregation mechanisms, how to form a presynaptic terminal, how to make that macromolecular complexes that can execute synaptic transmission, execute exocytosis and endocytosis. Along the way, we've also worked on dendrite morphogenesis by identifying receptor-ligand interactions that builds this very large, elaborate dendrite, as well as mechanisms of cytoskeleton polarization and membrane trafficking. And I won't talk for almost any of these papers, so that if you're interested, please stay after the talk, and we can get into that discussion. I want to focus on neuronal cell biology because neurons are extraordinarily large and complex cells with very long lifespan, right? So it's probably not going to be a hard argument to be made that neurons require specialized cell biology to accommodate this morphogenesis, to accommodate this compartmentalization, and to accommodate this very long lifespan of neurons. There must be very robust proteostasis and metabolic homeostasis to maintain these neurons to function for a long time. And most of the cell biological knowledge that we've learned comes from studies of Saccharomyces cerevisiae, the baking yeast, or from yeast 293 or HEK 293 cells. And we argue that you need to study neurons to really understand neuronal cell biology. Okay, so in the past 10 years or so, we've tried to establish an in vivo model using this highly arborized and highly polarized neuron in C. elegans as an in vivo model to systematically understanding different aspects of neuronal cell biology. And here, all the green structures are dendrites. It's a highly arborized and a stereotyped dendritic arbor, and with a single unbranched axon. So it is an in vivo system to study polarization, to study basic neuronal cell biology. And in this particular audience, I thought, the Department of Life Science here in Hong Kong University of Science and Technology has traditionally strength in understanding neurodegeneration. And I would make an argument that here, understanding neuronal cell biology is also going to be instrumental in understanding neurodegeneration. Imagine that we know that there are multiple mutations for the familial Alzheimer's or Parkinson's disease or any neurodegeneration diseases. The patients inherit a mutation from their parents, right? And this mutation essentially doomed the individual to develop early onset of neurodegenerative disease. But the clinical symptom would not show up until 4, 5, 6 decades later, right? So that argues that in the early stage, when the individuals are young, the cells must possess mechanisms, resilience mechanisms, to compensate, to mount adaptive changes to compensate for the mutation-induced cell biological changes. But over time, either it's aging or activation of immune systems or environmental factors or genetic predispositions, these adaptive reactions, responses, slowly becomes maladaptive responses that ultimately leads to terminal pathology. And I would argue that here, would be very helpful to understand how neurons deal with this stress. How are they, over time, what are the factors that actually makes these adaptive responses ultimately turn into maladaptive responses? Are there ways to stop the progression of the disease by understanding these responses and by manipulating these responses. So that will be the theme of today's talk. So in preparing for this talk, I thought I would pick some topics that represent sort of each of some from the very basic science in neuronal cell biology all the way to maybe disease mechanisms that are still relatively preliminary, but because Guojun is here, so I really wanted to pick his brain and other people in the audience with neurodegeneration expertise to get some feedback. So I'll start with microtubule organization, and then part 2 is neuronal stress response, and part 3 is cellular pathology, mechanisms of tau pathology. So okay, so throughout the talk, you'll be seeing this cell. As I said, this is a sensory neuron with is a very large cell. The cell occupies 80% of the worm body, and this is Nematode C. elegans. I forgot to say that. And then, so this whole animal is about 1 millimeter. So, and then the cell spans the length of 750 microns at least. So that's a pretty sizable cell, even comparing to vertebrate neurons. And of course, before it becomes mature, there are developmental, we can track the development, we can track the cell division that generates this neuron, and then it goes through a very stereotyped developmental process by extending axons. Always axon comes out first. There's no competition between axon and dendrite. The axons always deterministically shoot down, go to the ventral side, and then this is followed by the biogenesis of the anterior dendrite, that's followed by the posterior dendrite. And at L2 stage, you'll see that this is sort of a bare bone PVD with the anterior major processes, anterior dendrite, posterior dendrite, and axon. And later on, it branches into this very large arbor. So I'll be talking about microtubules. And I trust that the audience already know that microtubules are major cytoskeleton structures that are made of protofilaments that are made of polymers that are alpha-beta dimers, heterodimers. So these are the alpha-beta heterodimers are the bricks, the building blocks, and then they first form these long protofilament, and then 11 or 13 protofilament forms this hollow tube. And microtubules are major cytoskeleton structures. It's essential for basically motor transport within the cells. And the microtubules, once it's polymerized, each individual microtubule has a minus end and a plus end. The minus end is very unstable, and usually in cells is buried, is protected by something called the microtubule organizing center. And then the plus end exhibit dynamic behavior. You can observe the plus end behavior with a protein called EBP2 or EB2. So the EB2 is only recruited to growing microtubules. So I'll show you this very dramatic movie. So this is tagging non-neuronal cells with the EBP2 GFP. So here what you're seeing is that the microtubules are growing from the center of the cell outwards towards the periphery of the cell. So the plus end of the microtubules are all pointing to the periphery of the cell. The center of the cell is a structure called centrosome. Centrosome berries is where all the minus ends of microtubule, most of the minus ends of microtubules are. Is that, you guys following? Okay, good. So basically, I've seen a lot of students here, so I want to make sure that people are following. So, and then these non-neuronal cells are small enough, and that each individual microtubule could actually span from the centrosome all the way to the periphery. So the organization of this microtubule in non-neuronal cells is that there's a centrosome sitting in the middle of the cell, and then that jets off the plus end of microtubules. Okay, so that's the organization. For neurons, the situation is very different because the neurons are so long, right? We have axons that are as long as a meter. So the individual microtubules are about 10, 15, 50 microns at most. And then, so there's no way that a single microtubule could span the whole process. So the processes are occupied by partially tiled, staggered microtubules. Okay. But this creates a lot of problems for the neurons how to achieve this, right? For example, in the axon, here I've labeled the plus end of microtubules with green and the minus end with red. And in the axon of all animals, worms, flies, mouse, human, the plus end is always pointing to the distal axon. This is called plus and out orientation. And however, in dendrites, in the invertebrate system, it's traditionally thought that it's all purely minus and out. It's opposite direction of the compared to the axon. But in invertebrates, in mouse and humans, there are mixed microtubules. But the problem has always been that how do you get the minus and out microtubules into a thin process? Because plus ends can grow. Plus end is very dynamic. But the minus end, it's hard to imagine how it would grow. Okay, that has always been a problem. The solution is that the dogma in the field that is based on studies in cultured neurons is that there are short microtubules that are transported by, slided into the process through motor interactions with the existing microtubules. Okay, so I'll show you that we don't see that in C. elegans neurons, and we found a very different mechanism, and see whether you guys, how do you, so I'll show you the evidence for that. Okay, so for this neuron that I've talked to, PVD neuron, we know that from previous studies that we have done that in this main chunk of dendrite, the microtubules are all oriented in the same direction. So minus end is always pointing to the left side, and then plus end is to the posterior side of the worm. The axons is the opposite, is plus and out. Okay, so it's basically a one-way street in the sense that if you are a kinesin, you can go seamlessly from the tip of the anterior dendrite all the way to the tip of the posterior dendrite without getting confused. So it's all the microtubules are pointing to the same direction. We know that is the organization when the animal grows up, when it's mature, when this cell forms. Okay, so but the question is what is the developmental mechanism to make this happen, right? And each microtubule has an individual minus end and a plus end. How do they know where they are? How do they know whether to point this way or that way? Right? Each individual microtubule is its own entity. Like how do you coordinate this whole thing? I mean, that's a difficult. So again, we did it by this is Xing Liang, very talented postdoc in the lab, and did it by tagging, knocking in the GFP into the EBP2 locus. This is endogenous knock-in using CRISPR-Cas9, and you can see these spectacular movements, these comets. And then the way that we analyze these comets from, let's say, a developmental stage that looks like this. So the anterior dendrite has grown out. The posterior dendrite hasn't grown out yet. The axon, of course, is there already. The growth cone is here. That's the tip of the anterior dendrite. So we took movies like these. We converted them into chymographs. Chymographs is essentially a 2D representation over time. The y-axis is time. The x-axis is basically the distribution, fluorescence distribution along this process. So here, again, you can see numerous lines. Each of these lines represent a single microtubule that are growing out. Okay, so from a chymograph like this, it becomes immediately obvious. This was actually a big surprise. When we saw this, we were like, ooh, this is how the cell is doing it. And look at this is the entire cell at the time. All the microtubules are generated from one spot, which is this spot, right? So this spot is jetting microtubule out onto each side. And then, so that is the signature of microtubule organizing center. It's just like, it's a centrosome that is sitting in the middle of the cell in a round cell that's jetting microtubule out. But here, it can only shoot microtubule this way and this way because it's constrained by the plasma membrane of the thin tube, right? The thin dendrite. So here, but the big surprise is that this microtubule organizing center is not in the soma, but instead is behind the growing growth cone. By localizing this microtubule organizing center, it forces the rest of the dendrite to become minus and out. And that's the way it's made. And of course, again, this is a big surprise. It seems to be an obvious way to make minus and out microtubule in the thin tube, but no one has really proposed it because no one has had the resolution to look at it. So that's the power of direct observation in an in vivo system. right? So what is responsible for the microtubule organizing center? And then again, it's the usual suspect. It's gamma tubulin ring complex. This is the best studied microtubule organizing center polymerase, and it's responsible for centrosome microtubule organization. And here we knocked in a red fluorescence protein in the endogenous gamma TERK component GIP1, and then you can see that it's localized exactly to the place where the microtubules are generated. And there's only one microtubule organizing center here. If there's two, it wouldn't work, right? If it's two, then you have microtubule going both ways. And indeed, there's only one. And then for this to continuously work, the microtubule organizing center must also continuously migrate with the growth cone because the growth cone is growing, right? And so before that, is it important? Now, this is observation. Is it required? When we knock down this GIP1 molecule, and then you can see that the polarity of the microtubule is completely reversed, right? So now, instead of making the microtubule from here, now the microtubules are made from the soma, so it becomes plus and out. And then it doesn't move. So we, again, this is knocking, GFP knocking into GIP2, and then we're watching how it moves. Does it follow the growth cone? You see that this periodical jump only towards one direction, which is the growth of the extension of the growth cone. So, so it turned out that the gamma TERK is loaded onto the RAB 11.1 endosome. So the endosomes carry the gamma TERK and moving forward. So with these data and the data that I'm not showing you, we propose that the endosome carries the gamma TERK and then forms these clusters. And then these clusters doesn't know where they are. They just form microtubules around them both ways. And then the endosomes then moves on the distal microtubules using kinesin. And then in a new location, it'll form microtubules again. So through these round cycles of microtubule formation and migration, it builds the majority of the dendrite minus and out microtubules. Okay, so that basically summarizes. And another interesting question is how do you make them stable after you form them? And then Xing actually just published another paper, found an interesting mechanism that I won't talk about today. So this is the work of Xing Liang, a very talented postdoc in the lab in collaboration with Jessica Feldman's lab. So here, let's again take a look at this picture here, right? I mean, this is the prototyped, the first two processes. And then in the dendrite, in the anterior dendrite, there's a MTOC, and then that dictates that this part of the process forms minus and out microtubules. But what about the axon and the posterior dendrite, which has plus and out microtubules? Like there's no, you can't put an MTOC there because that would make this minus and out, right? And indeed, there's no MTOC from this end or from the axon. So what I'm about to tell you is that there's a different mechanism to generate microtubules in these plus and out processes. This is the work of a current graduate student, Dane Coano. So Dane took advantage of the microtubules in the posterior dendrite. It turned out that there's only a few microtubules in this very thin posterior dendrite, to the extent that he could label the tubulin. This is a tubulin GFP. So alpha tubulin, one type of alpha tubulin tagged with GFP expressed in this cell. And now what he does is he just takes time-lapse imaging on a very sensitive spinning disk confocal. And here's the movie that you see. Okay, you can appreciate that we can watch individual microtubules grow and shrink in vivo. So this is in a live cell, live animal, and the animal is still alive. It's not dead, right? So it's still polymerized and depolymerized. So you can see that. So in the past, this is only possible when you do this experiment on slides in purified proteins, right? So now we can actually watch, like in vivo, it happens. So with this kind of resolution, you can actually determine which mechanism is making microtubules. So there are there's de novo polymerization. Imagine you have a microtubule organizing center here. You can make microtubules this way, and we don't see that in this end. Microtubule sliding, the dominant hypothesis within the field, what you would see is a short microtubule that moves, right, that slides. And then in a chymograph, it would look like a band that moves diagonally because the length of the microtubules won't change. So that's the sliding signature. And then another behavior is severing. It's microtubule being cut. You have an existing microtubule, then next moment it becomes 2 microtubules. So this is severing. Severing, you would see these signature chimegraphs. So when Dane actually looked at the microtubules here and how does it actually, are microtubules generated in this process of development, and he didn't find sliding. He found very few de novo formation, and which makes sense because the de novo formation, you wouldn't be able to control the direction. It would be random direction. Okay, so he found severing. So here, the red arrows are pointing to 3 severing events, right? You can see that this is a microtubule, and then after this moment, it became 2 microtubules. Okay. The good thing about the severing is that after the severing, okay, after the severing, the orientation of the microtubule is preserved. So you have 1 plus end-out microtubule here. After severing, you have 2 plus end-out microtubules. Okay, this is a way to increase the number, make new microtubules from existing microtubules while preserving the directionality, the orientation. Okay, so these severing events this is actually the first time severing event has been observed in non-plant eukaryotic cells. Okay, so the plant people have seen them. But the severing because cells always have many microtubules, it's actually very difficult to watch severing in situ. But severing has been studied extensively in purified microtubules in test tubes. And then we know that there are enzymes that can sever microtubules. And these enzymes are super interesting. They're AAA ATPase. They're essentially chaperones. They're hexameric AAA ATPase. And then what they do is they land on microtubules, and then they pick up the C-terminal tail of tubulin, and they thread it through their ATPase, and then they unfold tubulin. Once the enough tubulin is unfolded, the microtubule will naturally be severed. So it's really not one of them is called katanin. It's like the Japanese sword, but it's really not a sword because the sword is more like, if you have a sweater, you have a loose end. If you start pulling on the loose end, you're going to have a hole very soon. So it's this mechanism is very well understood. Okay, each cell has multiple types of these AAA ATPase. Some you might have heard spastin, katanin. These are involved in actually developmental sensory disorders in neurons. And we found one that basically blocks the severing. So in a MEI1 mutant, there's no severing. And this is the accessory subunit of this MEI1 that also doesn't have severing. And MEI1 is the homolog, vertebrate homolog of katanin, which is one of these AAA ATPase. And you can see that the severing is gone in the MEI1 mutant. Okay, so nobody cares about weird dendrites that are plus and out microtubules. What about axon? So we looked at a different cell type, and indeed we found severing in distal axon as well. Okay, so that means this would be a very interesting way to generate new microtubules in a plus and out process. And the same molecular requirement in the axon compared to the dendrite. Okay, so if the severing is the main mechanism to generate new microtubules in the axon, you take it away, you take the severing away, does it lead to fewer number of microtubules? The answer is yes. Not super impressive, but indeed, all the severing deficient mutants have fewer microtubules. You can kind of appreciate just from looking at these chimegraphs, there are fewer microtubules in these two neurons. And does it affect trafficking? So here, when we looked at a cargo, this RAB10 endosomes, and you can see actually there's more static RAB10 in the severing mutants. And if you look at we can record these RAB10 vesicles in these processes, and then use chimograph to analyze their characteristic of their movement. And in the severing mutants, there's more pauses, there are longer pauses. We imagine this is because, the vesicles are being transported on a microtubular highway, and then they stop, they're waiting for the motor proteins to latch onto another track. And then with fewer microtubules, the pausing duration is longer. Okay. Okay, so where does the severing occurs? So here we plotted basically the relative severing frequency over the distance. The distance, the zero is at the very tip of the dendritic growth cone. So here you can see that almost all the severing events occurs within the first 30 or so microns. The majority of the severing occurs between, I'd say, like 10 and 30 microns. And then what is between 10 and 30 microns? Here I'm labeling actin and microtubule, and you can see the actin actually fills all the way up to the edge of the growth cone. So that's not surprising. We've learned a lot about axon and in morphology in actin and microtubule organizations in vertebrate cells, where the growth cone is always full of actin. The microtubule gets to the palm but doesn't really penetrate to the very tip. And this is exactly the same case here in the C. elegans neuron, which is the growth cone. The growth of the very edge of the growth cone is powered by actin polymerization, RP2/3 mediated branch actin formation. And the microtubules is essentially trailing the actin, the first actin zone. Okay, and then so essentially this is the perfect place to have severing event that generates more microtubules. Right? Okay, so it happens in the distal dendrite. Okay. So now we know that if the severing events are occurring at this distal event, what targets the severing? What actually makes this part of the microtubule more labile, or what targets the enzyme there? So in order to do that, we looked for other mutants that does not have severing. And we found two of them. One is called patronin, one is called NMTN1. So both of these looks just like the catenin mutant, the severing enzyme mutant that doesn't generate any severing event. So what are these molecules? These are very interesting molecules because they are known to be interacting with microtubules. Patronin is the vertebrate CAM-SAP. It's a minus end binding protein. It's known to stabilize the minus end of microtubules. NMTN1's homolog is called WDR47. That's also a microtubule binding protein. So these two genes are linked to catenin in a paper here where they showed that all three genes are required for the growth of the central microtubules in the cilia structure. Okay, so this is, but we don't know exactly how this works. So basically what we did is we made knock-in strains to label patronin and knock-in strains to label NMTN1, and then we asked where does the severing occurs. And it turned out that indeed, if you look at, sorry, this is the movie, and by the way, you can actually see severing. It's not very subtle. Like when severing occurs, you can see like it cuts into 3 microtubules, for example. So here there are multiple two severing events. And if you look at these events, so a new minus end is generated, and indeed, the patronin is here. A new minus end is generated, a new patronin is here, right? But interestingly, for this event, and even for this event, right, I mean, the patronin is there before the severing. So the patronin is already there. The supposedly 3' protein is already there before the severing event. So this actually argues strongly that it generates the minus end. Here is even more clear when we look at NMTN1, is that, for example, where is the severing event? There's a severing event right here, the severing event right here. But you can see the patronin is already there. So what these shows, and when we quantified the number of severing events that contains the existing patronin or existing NMTN1, the vast majority of them events contain these existing patronin and NMTN1. So this argues that when the growth cone is growing, behind the growth cone, there's signals that enriches these junctional proteins that are interacting with microtubules. And what they do is that they recruit the microtubule severing enzymes. And the severing enzymes then cleaves microtubules that generates additional microtubules to support the growth. As the growth cone is growing forward, right, and then it lays down these severing enzymes, and then that just locally generate microtubules to continue this growth process. Okay, so for this part, I mean, I think we were quite surprised by both of these mechanisms being, on one hand, very generic. They're using enzymes that we have known, there are structures for them. People have studied them to death in vitro. And microtubule gamma Turk has been studied in the context of cell division, spindles, mitotic spindles for decades. And we find that neurons repurpose these generic, quote unquote, generic mechanisms, but to tailor it to the needs for neuronal cell biology. So that's, I think, a lesson that we learned, that it's not like neurons actually invented brand new mechanisms. Instead, it's repurposed these generic cell biological mechanisms. And I guess the other thing that we are encouraged to continue doing is to take advantage of the in vivo systems. Okay, so I want to jump to part 2, which is, again, to understand how neurons respond to stress. And then this is really encouraged, or this is inspired by the notion that it takes so long for cell biological deficit to show up, even in mutations that are bound to make the individuals sick. So our approach is to take C. elegans neurons and use mutations to generate a stress condition. So we want to make the cell stressed. And then we ask the question, can we find suppressors, modifiers, that actually make the cell healthy again? And this is sort of like, if you have an ApoE4 carrier, can we find additional mutations to make the patient, to individual not have Alzheimer's disease or something like that, right? So it's to look for modifiers. And then in parallel, we could actually take vertebrate neurons, and then we could pharmacologically treat them to induce stress, and then use drugs or gene manipulations to ask whether these genes are resilient or not. Okay, so we know that from previous studies that the development of this very complex dendrite requires a lot of membrane proteins. Not surprising. You need the ER to work well. And then we know that in particular, because if you delete IRE1, this ER unfolded protein response gene, and then you lose the dendrite. Okay, so what is IRE1? IRE1 is one of the 3 pathways that underlies the unfolded protein response. So this is a universal cellular stress response discovered by Peter Walter's lab, that basically, if the cells are stressed, it causes ER protein unfolding within the ER. And then the cells have 3 ways, actually 4 ways, 3 depicted here, to combat the stress. The IRE1 or PERK, these and the ATF6, all 3 of them basically leads to more production of more chaperones, which then fold the protein better. Okay, that's the protective effect. But it also, these 2 pathways, IRE1 and the PERK, also shut down protein folding load, either by reducing the protein translation or by degrading the RNA. Okay, so when the cell is stressed, it doesn't want to fold a lot of protein. It produces more chaperones to fold proteins, and then it actually suppresses the production of proteins to reduce the stress. Okay, and then in parallel, there's something called ERAD, ER-associated degradation, which is a mechanism that recognizes unfolded proteins within the ER lumen and then threaded this protein, ubiquitinated protein, through this HERD1 molecule, which is a transmembrane ER protein, and then the ubiquitinated protein then is degraded by the proteasome. Okay, so if the protein is already unfolded, you can either refold them or you can thread it out of the ER with using this very specialized system and then get it degraded by cytosolic mechanism, which is the proteasome. Okay, so we know that the IRE1 is very important. It kills these dendrites. But can we really see, so in the absence of IRE1, we've generated a state where the ER is missing this major pathway, and then it's probably stressed. So we wanted to see whether we can find a protein to directly visualize the protein unfolding. And then here I have to introduce this molecule that we discovered, and this is a transmembrane protein called we named DMA1. It's a transmembrane leucine-rich repeat protein. We use this protein because this is a very meaningful protein. And we found it by doing genetic screens, trying to understand how these dendrite forms. And in DMA1 mutant, the dendrites simply don't form. So through many experiments, we know that this is the receptor molecule that is required on the plasma membrane for the dendrite to form. So this protein is important. So when we tag this protein with endogenous GFP, this is what wild type look like. In the IRE1 mutant, the protein is massively stuck in the ER. So the protein cannot be folded correctly, stuck in the ER, and there's not enough DMA1 on the surface, and therefore the dendrite would not form correctly. Okay, so this is our cargo, and then we generated a stress response. We also know that in addition to the ER proteins, endosomal proteins such as RAP10 is also required for dendrite morphogenesis. You mutate this RAP10 molecule, the dendrite is largely lost. Why is this important? When we look at the DMA1, remember the receptor protein, and then it's not only on the plasma membrane, it's also, this is the endogenous protein. And you can also see that a lot of DMA1 is in punctated structures in the cells, and those punctated structures are endosomes. So from a study that I won't have time to tell you, we know that the endosomes are required to recycle the DMA1 from the plasma membrane surface to endosome and then back onto the plasma membrane. So the continuous growth of the dendrite requires this continuous cycling. Okay, if you prevent this from happening, then the dendrites don't form. Okay, so now we think we have generated two different stress conditions, one with ER stress and one with endosomal stress. And here, just another way of showing this is that we labeled endogenous DMA1 and endogenous RAP7, which is a late endosome. And you can see that they don't colocalize because normally in wild type, DMA1 is largely in the recycling endosome RAP10 positive, not in the RAP7. RAP7 is the late endosome. That is a different type of endosome. Okay, but in the RAP10 mutant, you have a massive accumulation of DMA1 in the late endosome RAP7 positive compartment. So normally, RAP10 is recycling DMA1 back onto the plasma membrane, and in the absence of this major pathway, this cargo molecule is shunted to a different category, a different type of endosomes. And you can also see that the endosomes is already backlogged. There's accumulation of endosomes, like RAP7 late endosomes, beyond just DMA1 molecule, right? Here we're labeling all the RAP7 endosomes. So we have created a traffic jam by removing the RAP10. Now the RAP7 is accumulated. And then in the IRE1 case, this is very different. There's no accumulation, if anything, reduction of late endosomes, but a massive accumulation of DMA1 in the ER. So these are two very different cellular stress. And in the RAP10 stress, there's accumulation of late endosomes in the proximal dendrite, and in the IRE1 mutant, the protein are stuck in the ER. Not surprisingly, because we're manipulating very different genes. Okay, so the question is, can we find additional mutations that can reverse or rescue these phenotypes? So working with C. elegans, a traditional forward genetic organism, we can basically, what Caitlin did is she took these animals, she mutated them again, and looked for suppressors. So she found one, and this mutation, together with RAP10, rescued a good chunk number of dendrites. Okay, so this is still with complete knockout of RAP10. Okay, the RAP10 is gone. So this is something else that somehow rescues some of it. And then when she got this gene, and then she basically crossed the same mutation, SYN3, into IRE1 mutant, and it completely rescued the ER stress. Okay, and surprising, right? And then this gene, SYN3, loss of function mutation by itself has no phenotype. So how do we explain that? So, okay, so this is the dendrite phenotype being rescued. What about the accumulation of the cargos? In RAP10, SYN3, it's largely cleared the backlogged RAP7. And then very impressively in the ER stress, it completely cleared the ER traffic jam of this cargo. By itself, it doesn't have much of a phenotype. Okay, so these are sort of strange phenotypes that it took us a while to understand what it is. But of course, we can clone it. We identified what are the mutations, and it functions cell autonomously. And then SYN3 encodes a scaffolding molecule in a histone deacetylase complex. Okay, so it's an HDAC complex. And HDAC, this SYN3 scaffold HDAC enzymes and other histone binding proteins and transcription factors. These other factors target HDAC to particular genomic loci. Indeed, we found a single HDA3, which is a HDAC enzyme that also suppresses, as well as 2 other known members of the HDAC complex. So this whole complex can do it. So if you inhibit any of the components of this complex, it rescues. It makes the neurons more resilient to both of these type of stress. Okay, so HDACs are chromatin regulators, and this enzymatic activity leads to more condensed chromatin and repression of gene expression, right? So our hypothesis is that when you inhibit HDAC, genes are activated. There are additional genes that are activated, and these genes are what is responsible for the rescue, the suppression. And then we call these resilience genetic programs, okay? And then all of these are conserved molecules. So we can find vertebrate homologs. So this makes us wonder whether HDAC inhibition can protect vertebrate neurons as well. So to test this, we collaborated with Maya Meyernorf and Aaron Gittler's lab. And what they did is to treat corticoneurons, E16.5 corticoneurons with tunicomycin to induce ER stress. Okay, and then at this concentration, tunicomycin basically kills these neurons. And then, but co-treatment with tunicomycin with 2 different kind of HDAC inhibitors very dramatically rescued the death phenotype. Here, notice the broken arrow, and this is a very strong suppression. The way that we conceptualize these is that so normal neurons have IRE1 or RAB10 to execute protein folding or correct protein trafficking, so you don't need the resilience genes. But when you have mutations either on IRE1 or RAB10, the neurons are in trouble because they're missing these major pathways, and then they cannot compensate because the resilience genes were suppressed, were repressed, were locked away by the HDAC enzymes. So now if you inhibit the HDAC enzymes, now the resilience genes can spring into action and fix the problems. Okay, so that's our hypothesis. So of course, it is critical to understand what are the resilience genes, and are the resilience genes pathway specific, right? Is there the resilient genes that are designated to the ER stress versus the endosomal stress? And so to get at these questions, we did RNA sequencing experiment and a functional perturbation. So using mouse corticoneurons, we treated the mouse neurons, this is a wild type, and we treated it with tunicomycin or treated it with HDAC inhibitors alone or both. And then we sequenced, this is the principal component analysis, and you can see that these are orthogonal effect, basically. So there are many, many changes. There are thousands of genes that are modified by HDAC inhibition, not surprisingly. Others, like Li Hui Cai, has done these experiments a long time ago. And we can kind of conceptualize it as you can imagine that HDAC inhibition could have increased the beneficial adaptive response or inhibited the maladaptive deleterious response, right? Because there are genes that go up and genes that go down. And indeed, we can through these non-supervised cluster experiment, we could actually look for genes that are upregulated by the HDAC inhibition, and then maybe and speculate that they may be important for the beneficial response. And indeed, we found genes that are sort of make sense. But ultimately, so indeed, we found genes such as the other UPR genes that are further upregulated by HDAC inhibition, and but there's so many gene changes, expression changes, that I think you could essentially pick any genes or pick a lot of genes and then say, okay, I think these are the beneficial genes and those are the deleterious genes. But we really have to test them. So the candidates are so when we mutated RE1, and there are other ER stress pathways, right? ATF6 and PERK. These are parallel pathways. They can also do, increases protein folding load capacity, reduces protein folding load, and then the ERAD could degrade unfolded proteins from the ER. Okay, so these are strong candidates, but are they really the ones that are responsible? So we had to do the perturbation experiment. And so we went back to C. elegans, and then we created triple mutants. Basically, so here is the wild type, and RE1 loses dendrite, and then you have the suppression. Now this is, the dendrites are rescued. So from this background, now we individually deleted what we call the resilience genes, right? We are asking the question, which genes are required for the rescues to occur? Okay. So through this analysis, we found, actually, interestingly, ATF6 is required. You take away ATF6 as a triple mutant, now the dendrite completely fall apart. PERK is also required, slightly less, but still very much required. XBP1 is not required, and we can talk about that later. But other genes, such as the ERAD genes, these are the two ERAD genes, they're both required, and another chaperone cofactor is required. So we identified all of these genes. They fit the criteria of the resilience genes because they're required for the rescues to occur, and they're parallel pathways. Okay, and in the invertebrate systems, when we inactivated these two genes based on the worm results, it also killed the rescue effect. So what if we look at the protein folding, and indeed, when we inactivate any of the resilience genes, the protein folding went back to being trouble. Okay. But the biggest surprise is that as a single mutant, none of these resilience genes are required. Okay, so this is a real surprise because you could have said that these resilience genes, they're already making a contribution anyways. So if you take away some of these genes, of course it's going to make it worse, right? But that doesn't seem to be true, at least not at the level of single mutant analysis. So we can delete these genes individually, and they don't really matter. So this really argues that these are the ATF6, PERK, the other pathways of UPR, and the ERAD genes. They are legitimate resilience genes. They're normally not required because HDAC is inhibiting them. Normal cells really relies on IRE1. IRE1 does the lion's share of the folding. And then now, but when IRE1 is depleted or deleted or stressed, now these genes are required to come out to help. And all of them are needed. Right? Imagine you are a neuron that has lived in a familiar Alzheimer's patient's brain for some decades. And slowly, for some reason, this ER folding is problematic. If you can't simply reach in and have access to these other parallel pathways for some reason because of the chromatin inhibition, then you're probably in a worse situation than if you have the capacity to enlist or to use those. Okay, and I'm going to skip this a little bit because I really want to get to the third part. And the summary here is that we found that we also looked for the resilience genes for this RAP10 pathway. And that seems to be turned out to be different than these ER stress pathways. Okay, and so this is a different RAP, and then the RAP10 rescue does not require any of these ER genes. So that makes sense. These are two very different type of stress. Your backup pathways are actually individualized, but they're all under the control of HDAC. Okay. All right, so I think what we learned here is that there are latent resilience pathways, and then it has a lot to do with chromatin accessibility. And our hypothesis is that HDAC inhibition leads to open chromatin and enhanced accessibility to resilience genes. And there's actually a pretty sizable literature on HDAC being protective for neurodegeneration. This was actually beautifully shown by Li Hui many years ago with HDAC enzyme knockouts doing way better in forms of Alzheimer's models, as well as ALS, Huntington's. Okay, so I think what has really sets the field back a little bit is lack of mechanisms. The HDAC inhibition causes changes of thousands of genes. Like, how do we really conceptualize these differences? I hope these data helps us to sort of put them into a model that we can sort of digest it. Okay, so this is a part that really is preliminary. This is the work of very talented postdoc Yue Sun. And then she started about a year ago, and this has made quite a bit of a progress on this. So, and then we wanted to understand the pathology, cellular pathology of tau. So tau, of course, to this audience, you guys are probably very familiar, is a microtubule binding protein in normal neurons. But once hyperphosphorylated, it forms aggregate and ultimately drives the neuron, kills the neurons, and causes degeneration. And it's a major contributor, probably the killer for Alzheimer's disease, ultimately kills the neurons in Alzheimer's disease. But also the mutations are found in FTD, frontotemporal dementia, and as well as other neurodegenerative conditions such as Pick's disease. Okay, so we try to generate, so we actually didn't generate, Kramer et al. Generated a worm strain that expresses this mutant form of tau, human tau. It's a 4R, and that contains this mutation. The mutation is found in FTD. So, and we took this transgene and then we crossed it into our morphology marker, and we saw that indeed it's actually really messed up the neuronal dendrite. And it not only does that, so you can see that you can easily measure, I mean, there's a lot of profound deficit, but you can simply measure the anterior dendrite length as a proxy. But the phenotype is profound. So it causes disintegration of dendrite. It also causes quite a bit of a reduction in locomotion. So if you put worms in a drop of liquid, they will just thrash. And then you can count the number of thrashes per 15 seconds or per minute. And then the animals that expresses this human tau version, mutant form of tau, causes this. And reduction of locomotion. And then you can also, we tagged it with M-state gold, which is a very good fluorescence protein. If you guys haven't heard, this is the better version of GFP that doesn't bleach. Or bleaches very little. So we made an M-state gold single copy insertion fused with the tau, and you can see there's an age-dependent aggregation of tau in the soma of this large neuron. Okay, and so there are two seminal papers, one from Gan Li's lab and one from Martin Kempman's lab. They looked for genetic modifiers of tau aggregates. So they did this in iPSC-derived neurons, and then they used tau fibril in one of the studies, tau fibrils to induce the aggregate formation. They used disease form, antibodies specifically recognize the disease form of tau to do their screen. And then they bombarded the cells with CRISPR library to inactivate many genes. And they selected for genes that can modify the tau aggregation. Both of the studies converged on some weird proteins. Okay, so here on this side of the volcano plot are the genes, when inactivated, will reduce the tau aggregates. And these genes, UBC, UFC, UFL, these weird genes, they all belong to the same biochemical pathway. And both studies converged on the same set of genes. So this gene is called ufmylation, or someone said, some people say ufmylation, and it's you probably, how many of you have heard of this pathway? I know Guojun has. So because he has a paper on it. So this is a strange understudied pathway. Okay. So but these screens don't lie. They're unbiased. Both studies from 2 different labs, one in New York, one at UCSF. They all picked out multiple proteins in this pathway. Okay, and actually the studies where Guojun is part of it, it showed that the ulfilation pathway is also impaired in Alzheimer's patients' brains. So there are important questions that are still not answered. How does it work? And then what is the mechanism of tau aggregate? So why does it suppresses tau aggregation? And when you suppresses tau aggregation, does it really helps the neuronal health? Because this study, they just used the tau aggregation as an assay. So they didn't really assay the health. So now, since we have an assay, so we can test this. But first of all, what is ulfilation? It is a ubiquitin-like pathway that, through a series of E1, E2, E3 enzymes, conjugates this small peptide, ubiquitin-like peptide, UFM1, onto substrates. Okay, just like ubiquitination. Ubiquitin is conjugated on a particular protein substrate through E1, E2, E3 enzymes. Okay, but this one is much less famous than ubiquitination. Okay, so first we wanted to see whether this ulfilation pathway is also required for the worm tau pathology that we've seen. It would be pretty wild if it is, right? I mean, here we're hundreds of millions of years of evolution apart. Of course, this pathway itself is pretty conserved. You know, you look at the protein sequence, they're very conserved. Okay, so anyway, so we took our model and the tau aggregate model, and then here we have mutants that kills the activity or deletion of the E1 enzyme. This is the deulfilation enzyme. And indeed, they actually seem to suppress the tau aggregate. And then not only can we directly look at the tau aggregate, we can also look at the neuronal health. And here is the wild type, and then with the tau expressed, it messes up the dendrite. And then now, this is with the same transgene, but knocking out the UBA5. This is the E1 enzyme. It rescues the morphology. Okay. And it also rescues the thrashing to some extent. Not completely, but this deulfilase actually rescues the thrashing really good. So this is pretty remarkable. So, you know, it's a conserved, you know, I think, you know, this must be tapping into something really conserved. The biology is very much conserved. You put human mutant tau in worm cells, it damages the worm neuron in a way that utilizes something that is rather conserved and can be modified by the ulfilation pathway together. So the next big question is what actually, how does it work? How do you connect the ulfilation to tau pathology? So there are many possible ulfilation targets, just like ubiquitin. There are many proteins if not all proteins that are turned over by ubiquitin modification, right? So ulfilation also potentially have many substrates. But one particular substrate has been studied well. This is the 60S ribosome. I mean, that's the incredibly important structure. This is the structure that makes proteins, right? And out of the 60S ribosomes, there is a protein called RPL26. It's one of the many RPL proteins, ribosomal subunits. And this subunit is modified by ulfilation on these 2 lysine residues. So only lysines are conjugated by this peptide. If you look at these 2 lysines, it's actually very conserved. It's it's basically from C. elegans to chicken, mouse, human. It's dead on. Okay. So we made 2 mutations, mutating this lysine to arginine. So we preserved the charge, but killed the ulfilation. And then these mutants are alive, and then we know the RPL26 mutants are lethal. Okay, so work from Ron Capito's lab showed that the ulfilation of this subunit helps to kick the ribosomes off the translocon. So the ribosome sits on translocon on the ER membrane when it needs to make transmembrane proteins. And the ulfilation comes in here and then kicks off. And then his idea is that this helps to recycle the ribosomes for the next round of protein synthesis. Okay, So when we mutated this, the question is this responsible for or related to the tau toxicity? So here we made these very specific mutations. I just wanted to point out here that compared to the killing the ulfamylation enzymes, this is a very specific manipulation. Right? Here we've only changed the potential ulfamylation site on one substrate. And then when you kill UBA5, E1, E2, E3 enzymes, you kill all ulfamylation. Right? Okay. So this is a very specific manipulation. And that actually generated a suppression that is similar to taking away the entire ulfamylation enzymes. So you can see a suppression of the tau aggregates. You can see a or very strong suppression of the dendrite phenotypes, and also the locomotion phenotypes. So this really makes us believe that the ulfamylation pathway goes through the RPL, the 26, the ribosome pathways, and to resolve tau toxicity. So because the RPL26 is closely related to ER, so this prompts us to ask the question whether tau induces ER stress. And there has been literatures, including this paper from the Jackson lab, where Guojun is involved in, and that has suggested that the tau could be causing ER stress. So we wanted to do this, so we built a reporter, and then without getting into all the details into the reporter, we basically found that indeed this is the XBP1 GFP reporter, and in the wild type it doesn't get turned on very strongly, but with the tau transgene it turns on very strongly. And strangely, when you actually take away the ulfamylation pathway or the RPL26 mutations, this very strongly suppresses the ER stress. So this tells us that the tau seems to cause the ER stress, and then the ulfamylation pathway seems to be downregulate the ER stress. But in this case, is the ER stress good or bad? Is it protective or is it detrimental? Right? Because we have these functional assays, so we can ask these questions by deleting the ER stress genes. But before that, so one problem is that this tau is in the cytosol and ER stress is inside the ER lumen. They're separated by the ER membrane. How could this be, right? So, but one idea is that it could be that the ERAD, the ER-associated degradation, is what links these two, because ER-associated degradation is threading out the unfolded proteins from the ER to be degraded by the proteasome, which is in the cytosol. Right? So to test this, we actually devised a ERAD sensor. This is a protein that contains mutations that causes protein unfolding inside the ER. But when we co-express it with the mutant tau, and we found that the ERAD sensor is actually co-accumulated with tau in the cytosol. So that argues that they compete for, the tau is competing with ERAD for the capacity of the proteasome, and that is probably how it causes the ER stress. Okay, so this is the last slide, and we found that deleting the ER stress genes are actually beneficial, are protective. So this argues that this particular ER stress that in the model that we have established seems to be maladaptive and detrimental. Okay, so here what we learned is that we think that tau is causing an ER stress, probably by competing the capacity of proteasome with ER-associated degradation, causing ER stress protein unfolding, and this causes a maladaptive response that causes toxicity. And the ulfamylation pathways through RPL26 inhibit this ER stress, and therefore resolving the tau toxicity. And then this is obviously still preliminary. There's a lot, we can do a lot more. I think we have the means to actually, the ways to do a lot better. So I think I've mentioned most of the people during the talk, and Xin Liang is responsible for the microtubule work. Then Kwano is isolated the serine enzyme. Caitlin Taylor is responsible for the stress response, and USON has been making progress on the tau toxicity. I want to thank my collaborators and then funding sources. Thank you.
Any questions?
Online? I'll repeat the question if you.
Here.
Very nice talk. I just have a question about the first part, about the microtubule severing. So how you choose the site of severing? I guess is the severing, does it happen at random sites on the microtubule, or are there particular tubulins that are more prone to
I see.
unfolding? Yeah.
Interesting question. I don't think it's, I'm sure it's known. I don't know the answer. I don't think it distinguishes tubulin isotypes. So each organism has multiple alpha and betas, and the vast majority of the experiment that has been used to study this is from cow brain tubulin. So when you study microtubules, you're basically going to the slaughterhouse, and then you're taking like a big cow brain, and you grind it up, you go through these polymerization, depolarizations procedure, and you ended up with microtubules that are made of multiple type of tubulin. And then, I don't think it's isotype specific. I'm sure it's known whether it's alpha or beta. I can't tell you. I could be wrong. Yeah.
But just in terms of the position on the on the microtubule, it just
no, it's random. It's random. It's really interesting. This is a work from Antonina Rowe-Macaque who was trained with Ramvale, and now it's at NIH. So what she showed is that in vitro you can add so low concentration of these catenin, and then what you see is that you're going to plug some holes, and then there are microtubules that can actually go. The free tubulin can go in there to fix it. That was actually the first time people showed that incorporation of tubulin does not necessarily have to be at the ends. It can also happen so it's randomly distributed, lands on the microtubules, and then it just unfolds it. Yeah.
Thank you for the wonderful talk. I also have a question for the first part. So I'm just curious about the functional significance of the microtubule severing in exons. And also second question is, once the microtubule undergo severe, what is the fate of the microtubule?
Yeah.
And is there any gamma tubulin associated with the minus end?
Yeah, great question. So I'll answer the second question first. So when the microtubule is severed, you've generated a new plus end and a new minus end, right? And we've seen situations where the minus end is stable. Then we've seen in many cases the minus end also disappears or shrinks, okay? And then the plus end is usually shrinks first. So we estimated 1 out of 15 to 20 microtubule will survive. And most of the microtubules will disappear. So it's a very inefficient way because microtubules are very dynamic. And I think it seems to be a very inefficient way to make microtubules. But it's actually compared to the MTOC, the microtubule organizing center in the anterior dendrite, it's actually not very inefficient. But because if you look at the MTOC in the anterior dendrite, it generates 30 to 50 microtubules per minute. So it's jets off microtubules all the time, but only like a few percent, less than 1% will survive. So microtubules are very dynamic, intrinsically dynamic structures. And then the MTOC is there to ensure that directionality is correct. Because it's actually, there's some sort of profound, interesting thing about it. And so this MTOC is actually, I forgot to say, is a developmental structure. It only exists during the extension of the dendrite. It disappears when the dendrite reaches its destination. And these microtubules are then preserved for the life of the cell. And imagine if the microtubule actually completely disappeared and then it wants to reform again, it will have no idea to know which way is what. These are molecules, right? They don't know which way is distal, which way is proximal. So it becomes sort of random. So you have to have like this very dynamic system, and then to ensure its directionality, and then a lifelong maintenance program to keep it from like disappearing. So that was our answer. So your first question is, what is the impact for the axon? Great question. We're still working on it. I have to say that the reduction of the number of microtubules in the dendrite that we've quantified so far is underwhelming. It doesn't seem to be a tremendous drop. And I think the dendrite is short. So, you know, the novel microtubule growth can kind of cover it. So the experiment that we're doing now is to look at long axons and look at whether there are delays or whether there are, you know, microtubules just won't get there fast enough to have serious troubles. So that is a very, you know, I'm still undecided on how important this thing really is. But we really don't see any sliding. So it's not microtubule sliding. And you know, vertebrate neurons could have this complex called augmin. Augmin is a polymerase that can make microtubules along existing microtubules, but worms don't have those, and each worm axons have plenty of microtubules. So I'm sure there's a mechanism. I think severing is likely to be one, but I think you're right. That part needs to be strengthened. Yeah, thank you.
Yeah, I have a question. First of all, great talk. So I have a question regarding the HDAC part.
Yeah.
In cells, there are different types of histone deacetylase. So have you ever tried to inhibit other type of the histone deacetylase rather than just SYN3?
Yeah. So in C. elegans, we tested all 7 HDAC enzymes. So there are mutations in each of them. So we generated mutations and use the existing mutations. There are 2 HDAC3, which belongs to the class 1 to 3 HDACs. So these are nuclear HDACs. You can't tell whether it's 1 or 2 or 3 compared to the vertebrates, okay? But it's one of them. So HDA3 showed a beautiful suppression, as I showed you. There's a HDA1, which is another type 1 to 3 HDACs. So that one has a strong developmental phenotype. They're cell-fate defect. So I think these 2 might be the ones that actually makes a contribution. I don't think it's HDAC6. Lots of people ask the question whether it's tubulin HDAC. I don't think so because of all the other cofactors of the nuclear HDACs that also plays a very important similar role. So I'm pretty sure it's in the nucleus. I'm not sure. You know, the enzymes, the inhibitors that we use, TSA or piscidine, are relatively broad spectrum, but still focusing on the HDAC 1, 2, and 3. So those are the nuclear HDACs anyways. Yeah.
Very nice. I'll have 2 quick questions. The
Yeah.
first one, for the first part, do you have any clue what's the factor to determine whether the mTOQ is formed or not at the growth cone? Because there could be several factors like diameter, length, complexity, orientation, even timing.
Right. So the mTOQ, that's a great question. So we actually, this is something we haven't published. And so basically we're looking at the transition between cell division and initially how the mTOQ gets into the anterior dendrite. There's only one. And so during cell division, there's the mitotic spindle is chock full of mTOQ. It's 2 huge cluster. Okay. And then post-division, one disappears. So sorry, the becomes much dimmer, and then the microtubular aster moves around, and magically gets targeted to the ventral side in the place where the dendrite is about to form. So we know that that behavior requires NUMA, which is a spindle localization gene. It requires a Wnt pathway. So basically, there are extrinsic cues, morphogenetic gradients that defines the site of the dendrite formation. And then there's a set of another generic set of factors that are known to move mitotic spindles in asymmetric cell division, by connecting the spindle microtubules to the plasma membrane, utilizes those mechanisms to drag it there. So again, that's a great question. I think those are basically, it probably happens in every cell, every neuron that it will be generated. Okay, second question.
Second quick question is whether HDAC inhibition allows or permissive ways to allow multiple stress response pathway to be like coping with the existing ER stress.
Yes.
Or alternatively, itself is a stress that activate those stress pathways and then fix the ER stress as a side product.
Yes, okay. So this is getting into some really interesting things. So okay, what exactly is HDAC inhibition doing? Okay, I think without getting into too much details, I think it actually reverses development. I think the HDAC inhibited state is a juvenile, young sort of state. So that state actually has less differentiated, allows like the cells to be in a state that is not very committed to being a neuron. But it actually didn't change the cell morphology. So we're doing more tests on the function of this neuron to see how bad, what is there like a high price that you pay to inhibit HDAC? Yeah, that's a very legit question, good question actually.
Thank you, Professor. I'm wondering if it's possible that the human mutant tau would cause damage of organization of microtubule so that cargo cannot transport?
Yeah, so that's actually, initially, that's what the field thought, right? Is like if the tau comes off microtubules, I don't think it has been supported by strong evidence. You can make a tau mutant, and it doesn't really actually have strong microtubule phenotypes. There are many redundant microtubule-associated proteins. So we actually found one that is very important for microtubule stabilization organization. That's not tau. So I don't think there's strong evidence that tau coming off microtubule has a profound impact on the microtubule. Yeah.
So actually my question is somewhat related to that, the tau toxicity. I think the functional role of tau, that field is coming back a little bit, which is loss of function, right? So if you don't have enough tau to stabilize microtubule. But my question is more related to the tau toxicity. So obviously, I think the field all in general agree, tau pathology or tau is killing neuron. And I think you said that and correlate really well with pathology and the clinical outcome. I guess the question is, the field really doesn't know what form of tau and that kill neuron, how does it do it? So recently there's a Cell paper, I think Brian Hyman's is the author, that they show tau oligomer actually could be the culprit for the toxicity. So I guess in your case, obviously this particular worm model, it does generate this cellular toxicity, but you use the aggregate primarily as a readout. And I think one of the slides you have a question, how did these aggregate actually cytoplasmic domain trigger ER stress? And I wonder, morphologically, did you look at a higher structure, whether there's a physical interaction between this aggregate and morphology of ER? And also beyond the toxicity, are there other readout toxicity, such as the behavior of this worm?
Right. So we do, okay, yeah. So I think at this moment we're ambivalent in terms of, so when we express tau, we do see diffuse component, what we call diffuse component in the cytosol. We do see actually microtubule binding like structures. Actually, it's actually, you can actually see them. Actually, is this clear? Like I feel like when in a wild type, when you see a lot of aggregates, you actually you started seeing these tracks. You definitely see microtubule stainings. I mean, these tracks are microtubules. But what I would say, whether this diffuse component is the oligomer that is detrimental, I don't know. I think we do see aggregates, we see microtubule bound forms, and then we also see potentially oligomer forms. So I mean, we do have these we have the neurite integrity as an assay, right? This is, again, is independent of which form is toxic, right? And then we also have the thrashing assay, which is also independent of which forms. Yeah, I really wanted to use some of the antibodies. I mean, are there oligomer-specific antibodies?
There are some antibodies. I wouldn't say that they're oligomer specific, but some form of phospho-tau might be more specific to either aggregates or oligomers.
That's right. So I think
well.
I think those would be the experiment that we need to do to. That's a good question. And are there mutations? Yeah, we should talk more about this.
Yeah, I mean, maybe this soluble oligomer could be sequestered where these insoluble aggregate could not, and that could be an angle. I think the field of thinking about the therapy or reverse the tau. Really toxic.
Good point. Thank you.
One last question.
Okay.
Hi, Kang. This is Li Ting. Nice to see you again in Hong Kong. So 2 quick questions. The first one is the unique position of MTOC. Did you find similar position in other mammalian systems?
Yeah, so we are trying. So I think that the equivalent stage of this is during the migration of cortical neurons on the radial glia cells. And then, so in that migration process, the leading edge will become the apical dendrite. So that's already sort of determined at that stage. And so I think that is the right stage, and no one has looked. So we're actually working to look at that. So Adrian Moore's lab in Riken Institute in Japan, they looked at Drosophila, found a very similar microtubule organization in the sensory dendrite of Drosophila neurons.
Okay. So second question is, although in C. elegans there's no neurofilament, but in the intermediate filament still exists. So in this case, did you observe any correlation between intermediate filament together with the spatial dynamics of microtubules you observed?
No, I don't know why do you have any reason to think that intermediate filaments is important?
Because the neurofilament dynamics is also associated with microtubule. So
they're much less dynamic than the microtubules, right? And then in vertebrate cells, they thought to be like filling the space and then really building sort of the large axons that has a lot of intermediate filament. That's interesting. We haven't looked. Yeah, maybe we should take a look.
They are transporting.
They're transported. That's right. That's right. There's Anthony, whatever his name, from Ohio State has done that for a long time. Yeah, great. Thank you.
With that, there's a reception on the 5th floor living room. Please join us Suzanne for a great lecture.